G'day everyone, welcome back and thanks for joining me. Remember that before we get too deep into the weeds regarding the historical impact of pharmaceuticals, we want to spend a couple of episodes laying down a basic scientific understanding of how drugs produce their effects on the body. And obviously for that to happen, the drug actually has to get into the body in the first place. And so we talked about in my earlier video the important processes that occur during the so-called pharmaceutical phase of drug action. And this begins when those carefully formulated and manufactured pills are ingested, they're swallowed by an individual, they begin disintegrating and dissolving, and this eventually produces smaller and smaller particles that fully dissolve, and eventually we get to individual drug molecules in solution. And this is when pharmacology know-how and expertise takes over from the pharmacists, and we begin to talk about those fundamental processes that shape the pharmacology of drugs, one of which is their pharmacokinetic properties. And remember, we talked about pharmacokinetics being what the body does to the drug, and it's controlled by four fundamental processes which are indicated by the acronym ADME, and we're looking at the first one today, A for absorption. And it only occurs when we have individual drug molecules swimming around in the fluid within the gastrointestinal tract. And remember also we introduced the all-important concept of a therapeutic window, and this is based on our knowledge of drug concentrations in the blood as a function of time, how they change, how they rise after a patient ingests some pills and reach a maximum and then begin falling as the drug is removed from the blood. Okay, so essentially what we're talking about today is this process of absorption, and essentially it has to do with, it strongly determines how quickly blood concentrations rise as well as the maximum peak height that they reach within the bloodstream. And remember, this is really important because if you go back and look at my earlier video, we want to give doses of drugs that remain within the therapeutic window for patients, the healing window we might call that. If we go too high, we'll start causing toxicity, harm to patients, and also if we give too low a dose, they won't even begin to benefit from them at all. So there's a sort of sweet spot we're trying to achieve and maintain when we're treating patients with pharmaceuticals. Okay, so the first process then is important in determining how well a patient responds to a medicine is absorption. How well is the drug absorbed into the body? Okay, so this essentially refers to the fact that from a pharmacokinetic point of view, the body can be conceived as just a sort of walking digestive tube. Okay, it's a pretty reductionist view of humanity, but essentially from the point of view of getting drugs into patients and people, that's basically how we need to think. And we understand that the digestive tube is not where the drug needs to go to produce its effect most of the time. We want the drug to get out of the digestive tract contents and into the tissues of the body. Okay, so otherwise it'll just remain in the GI tract and eliminated via our feces. Okay, so absorption is the process whereby a drug enters the human body, that is, whereby those drug molecules that are cleverly packaged within the pill, that payload, chemical payload, is released from the pill and crosses the barriers that surround the digestive tract and enter into the bloodstream. Okay, so it's an all-important process. If a drug isn't absorbed, then it won't produce any benefit, any change in physiological functions within the body. Okay, so today we want to look at the key factors that influence the readiness with which orally ingested drugs taken by mouth, move from the gastrointestinal tract into the internal body environment. And this is fundamentally important in pharmacology. The better a drug is at doing this and more consistent and reproducible it is, the more stable that drug will be in terms of predictable effects in patients. Okay, now we're going to focus on the use of drugs orally in this lecture because that is the one that's most important most of the time. If we wanted to be precise about it, we'd also look at some of the non-oral routes of drug administration. But we're going to focus on the use of oral administration because that's simply the most convenient way, easiest for pharmacists to send off patients with their packet of pills and instructions to take the 2 pills every 8 hours with a glass of water. A very simple, convenient way. But it's also favoured because it's the safest way. And we're going to look at some of the protective capabilities within the gut wall and within the liver that mean that the body detoxifies and protects itself against chemicals entering the body. Okay, so it's usually safer and a less risky option to take a drug via the oral route than it is to, say, inject it into a blood vessel or inject it into muscle or subcutaneously under the skin. Okay, so this process of drug absorption fundamentally occurs after oral dosing from the upper small intestines, and that's the part of the gastrointestinal tract that basically connected to the stomach, the bottom of the stomach. We call it the duodenum, and that's where most drug absorption occurs. Some drugs also get absorbed from the stomach to some degree, and a few also might get absorbed significantly from the large intestines, the colon, lower down in the digestive tract. But most of the time, most drugs get absorbed from this so-called upper small intestine known as the duodenum. Now, what happens then is much the same as when we eat a meal and all the proteins and fats get broken down into their constituent molecules, releasing lipids and fatty acids and sugars, small carbohydrates. The same processes that absorb those nutrients into the body via the bloodstream are essentially the same fundamental processes that introduce drugs into the body. So we're not talking about anything too magical when we talk about oral drug administration. Now, essentially what happens is the molecules, if they have the right properties, are drained via this so-called portal blood vessel, this portal vein, which essentially drains molecules from the gastrointestinal tract and carries them to the liver, the first port of call for chemicals that enter the body. It's very important that that happens because the liver is amazing at protecting the body against chemicals. Okay, so when we talk about drug absorption, we're really talking about the readiness with which drugs transfer from the interior of the upper small intestine, the duodenum, into the bloodstream, known as the portal circulation, the portal blood that's carried by this major vein within the body from the gastrointestinal tract to the liver. Now, it's important to appreciate that not 100% of an absorbed dose necessarily gets into the bloodstream, and this is what we're going to talk about, why some drugs get in better than others. That's really very important in pharmacokinetics. Now, essentially, we won't go into the science too much at all because it can get pretty complicated, in fact, very complicated, and you need a good background in chemistry to appreciate it. We want to sort of fly across this topic at a height of 30,000 feet in these lectures. So we want to keep the science as simple and accessible as we can. But fundamentally, there are three processes whereby small molecules can get across the tissue barrier, the interior lining of the digestive tract of the small intestines. Okay, so this is what we're talking about in absorption. Molecules need to get out of that intestinal fluid and across this cellular layer, barrier of cells, and get into the bloodstream to make their way to the liver. And the first process that's being recognised is so-called paracellular division. Sounds like a big term, but para means next to, and essentially it's molecules getting into the bloodstream through gaps between the adjacent cells in this intestinal lining. Okay, so this is generally only relevant for small molecules, very, very small drugs. So a good example of an everyday chemical that gets into the body this way would be alcohol. It predominantly is absorbed into the bloodstream by so-called paracellular diffusion. Now, when I was a boy learning pharmacology well over 30 years ago, we thought the main process was passive diffusion. And that passive diffusion means that the drug goes through the middle of cells, and it means that it has to be able to get through these blue and yellow structures here that I've shown, these outermost cellular membrane of cells. And this is called transcellular diffusion, and essentially means that the drug is just going to diffuse without any energy input from a high concentration environment within the gastrointestinal fluid into a low concentration environment in the blood. It's just passively drifting along down its concentration gradient, and it's making its way through this cellular barrier. Now, that's a challenging feat because that cellular membrane barrier is essentially defined by what we call the lipid membrane or cellular membrane bilayer. And when we zoom in and look at what that's made of, we're not going to the chemistry at all, but essentially these phospholipids, these modified fatty acids, are used in the manufacture of the cell membrane. And they have a water-loving head, a hydrophilic head, water which prefers to be dissolved in water, and then connect, that's shown in blue, but then there's this they're connected to this hydrophobic tail. This is essentially hydrocarbon tail of a fatty acid. And these are very greasy, water-hating species. So essentially what happens, and this is essentially what you see when you're working in your sink and you have grease coming off, you've been frying something in a pan, and you see, once you put the greasy pan in the water, you see all these little droplets forming. They're spontaneously forming these sorts of membrane bilayers where the greasy droplets have structures, form these spontaneous structures with a hydrophilic head orientated outwards and the greasy tail inwards. So this basically means then that for a drug to be able to get through the cell in this sort of transcellular diffusion, passive diffusion manner, it needs to be compatible to some degree with that water-loving head, but also it has chemical properties that allow it to penetrate that greasy, water-hating tail. Okay, so this is one of the challenges in medicinal chemistry, designing drugs that have a balance of these two properties, because they're the best sort of molecules that get passively absorbed. Now, so that's passive diffusion, transcellular diffusion through the middle of cells. But we now know that one of the really important determinants of how well drugs get absorbed into the bloodstream from the gut also has a role to play for some drugs, at least, with these tiny molecular turnstiles. I'm going to come back to these later in this session. But these are tiny little pumps that essentially assist the movement of small molecules across lipid cell membranes. And we call this facilitated diffusion or aided or pump-assisted diffusion, and these tiny structures are very important in facilitating the movement. Now, they're pretty selective, so particular pumps tend to prefer particular types of molecules in terms of allowing them into the cell. So they're a little bit like an exclusive pub. They don't just allow any old riffraff into the exclusive club within the pub. They only allow certain types of characters. And so in the same way, different types of membrane pumps are all located around the body in all sorts of tissues, and they have a lot of say over which molecules get into tissues and which don't. We'll come back to that in a minute. Okay, so there are three fundamental processes that can shape the readiness with which drugs get into the bloodstream. Passive diffusion is very important, but also for some drugs there's a very important role for these membrane pumps as well. Now, that's all very interesting from a molecular and cellular sort of point of view, and researchers like to study those sorts of phenomena if they work in research laboratories studying all those different pathways. But from the point of view of using drugs in a patient, we really want a simple way of determining how well does a drug get into the bloodstream. So this is where we use this concept of oral bioavailability. Bio means biological, available just means how readily is the molecule in where it needs to be in the body. So oral bioavailability just refers to the fraction of the oral dose that reaches the bloodstream. Okay, we also call this the F-oral. And how do we determine this? Well, remember how I talked about in my earlier video the all-important blood concentration versus time curve. Now, before I talked about the importance of understanding the speed at which the blood concentrations rise and fall, and also the size of the peak, where that is in terms of whether it lies within the therapeutic windows, whether or not patients benefit. That's all very important, but just to add a bit more complexity to the value we can extract from a blood concentration versus time curve, I want to explain a new concept, and that is the area under the curve occupied by blood concentrations of drugs within the body. Okay, this is really important in clinical pharmacology because you see the size of the effect that a particular dose of a drug produces in a patient is often more closely related to this area occupied by the curve, under the curve, rather than even the maximum concentration. Not always true, but quite often the area under the curve is a particularly valuable indicator of how much drug is really getting into the body after dosing a patient. So that's really valuable in clin pharm. Now, the interesting thing, though, to determine the fraction of the dose getting into the bloodstream, we also do a second study. So we send our volunteers home and they come back a week later and we give them a second dose of the drug, not via the oral route in this case, but now we're giving it via a direct injection into the vein, via an intravenous dose. Okay, so again, we take blood samples and send them off to the lab, and they measure the concentrations of the drug in the bloodstream after this intravenous injection of the drug. And essentially, we can plot the data, and we get a different type of curve. And because we're directly administering the drug straight into the bloodstream, we get the highest blood concentrations immediately after injecting the drug. So this is why heroin users like to inject heroin. They get a rush and that's because they get this bolus of drug, high amount of drug directly getting into the bloodstream immediately. There's nothing like the liver or the gut wall slowing it down or metabolizing it and protecting the body. It's all going straight into the bloodstream. Okay, so we do the same thing in clinical pharmacology, but we want to estimate the area under the curve via both the oral route, shown in orange, and then the intravenous route, shown in blue. And essentially, the fraction absorbed is simply the area under the curve, the orange part here, this area, divided by the area under the curve under the intravenous dosing. And this tells us this all-important fraction of the administered dose that enters the bloodstream after oral dosing. Okay, so by definition, with intravenous dosing, that F is 100%, because as you can imagine, it's completely bypassing all the anatomical structures within the body that impede the entry of drugs, the gut wall and the liver. Okay, so this is how we calculate the fraction of the absorbed dose, that oral availability. And just to show you how it varies for common drugs, ibuprofen is, of course, we're all familiar with this anti-inflammatory analgesic drug. It's probably one of about 10% of most of the pharmaceuticals we use that have 100% availability. That is, all the ingested drug gets into the bloodstream. On the other hand, another analgesic drug, a more powerful one, codeine, only about 50% of the ingested dose actually makes it into the bloodstream because it gets metabolized extensively within the gut wall and the liver. But then there are other drugs where we have even much lower availability than that. So a good example would be mercaptopurine, a drug I hope none of you ever has to take because it's used in the treatment of serious conditions such as leukemia. But mercaptopurine has a low oral availability of only about 10%. Now, sometimes this really matters, and we need to understand why drugs have low bioavailability, because sometimes it can help us choose other medications when we're treating patients with multiple medications. We don't want drugs interfering with whatever process causes low oral availability in a patient for a particular medicine, particularly a toxic one like mercaptopurine. So that's all beyond the scope of this introductory video, but maybe in the future we'll come back and look at some of these processes in more detail. Okay, so you might be wondering, why is it that less than 100% of the dose it gets into the bloodstream? Well, essentially what happens is it's the presence of these all-important gut wall and liver, and these are there to protect the rest of the body against invading chemicals entering the body. Okay, so if you can imagine, when we give 100 milligrams of a drug, let's say, shown here in these little yellow molecules, we're giving 100 milligrams via the oral route, and we can imagine that perhaps not all of that ingested 100 milligrams actually gets absorbed. Let's say only 90% of it does, and the remaining 10 milligrams is unabsorbed and excreted in our feces. Okay, so that's already reducing their availability. But then, more importantly, for most pharmaceuticals, they often undergo metabolism by enzymes in the gut wall, but also after they make it through the gut wall, they can get carried by the blood to the liver, and there's a lot of metabolism going on. And essentially I've shown that here with these new structures here. The drug has now got these OH groups, these oxygen and hydrogen, and these have been stuck on the drug as they make their way through these cells in the gut wall and the liver, because enzymes in these tissues are able to recognize that foreign molecule and think, oh, we don't want you around here, we better convert you into something that the kidneys can get rid of and make you more water soluble, so we'll stick an oxygen and a hydrogen on you to make you more water soluble. We'll talk all about that in a future video. Okay, so let's say we're losing 20 milligrams through these combined processes of metabolism in the gut wall and the liver. So that means that we've now got, in terms of the amount of ingested drug that can make it into the circulation and reach those other body tissues, of just 70 milligrams. Okay, so now the oral availability as you can see, is 70% or 0.7, a fraction. 70 divided by 100.7. So just 70% of the ingested drug is getting into the bloodstream due to the combined influence in this hypothetical example of incomplete absorption and metabolism within the liver and gut wall. Okay, now for the rest of the lecture, I just want to quickly zoom through at our 30,000 foot elevation and just look at some of the factors that can cause less than 100% availability for drugs. And this is the sort of stuff that's really important in pharmaceutical science, and the people who work on optimizing oral absorption of drugs spend their whole careers thinking about these issues and trying to optimize it. I'm just taking a really superficial zoom over the top of the field, so please forgive me if I'm not going into the chemistry the way I really ought to. But essentially, one of the reasons why some drugs have lower oral availability is that they're chemically incompatible with the really harsh, strongly acidic conditions within the stomach. And I'm sorry, this should say stomach up here rather than intestine. But essentially, we all know that the stomach contains very high concentrations of hydrochloric acid. And it's there because it helps prepare food for digestion and extraction of nutrients from the once it reaches the small intestines. But essentially, and also it's there to kill off any pathogens, any microbes and bacteria that might be in our food that we eat. This is the first level of defense against a lot of foreign substances. But it also can kill off and destroy any acid-sensitive drug molecules. A good example of a drug like this would be some of the penicillins that are notoriously acid sensitive, and they simply undergo breakdown in that strongly acidic environment, low pH environment. Okay, so in this case, low oral availability for these sorts of drugs can occur because very little drug survives that harshly acidic environment within the stomach. Okay, so we can get around this, and formulation chemists are very good at making enteric coatings and putting materials around the surface of the pill so that the drug survives passage through this strong acidic environment. But we'll come back to that sort of issue in a minute. Another reason for low oral availability is that the drug might actually be too big. And again, this is a complicated topic, but essentially we calculate the molecular weight of drugs by counting the number of different types of atoms, the number of carbon atoms, hydrogen atoms, oxygen atoms, nitrogen, sulfur, chlorine, fluorine, and each of those elements has its own atomic weight. And we count them up and multiply that atomic weight by the number of times an atom is represented in a drug. We add all that mass together and we get the total molecular weight, the weight of the whole molecule. And essentially, if we want a drug to be well absorbed and to have a high FO, high fraction oral absorbed from an oral dose, it's got to be not too big. It's got to have a total mass in terms of grams per mole of less than 500. If it's bigger than that, we can start having problems with poor, unpredictable absorption. Okay, these are not absolute hard and fast rules, but they're just sort of general rules that the pharmaceutical industry has been following at least for the last few decades. That if you want to maximize the chance of a drug getting into the body from the oral route, you want to stop it from getting too obese. You want to stop it from getting too big and bulky. Another related problem that is really important in determining how well the drug gets into the bloodstream is the balance of its solubilities and whether it's greasy or grease-loving or water-loving. Now, the chemical terms we use for this, so we're really thinking about the fundamental chemical and physicochemical properties of the drug molecule itself. On the one hand, we want the drug to have a bit of greasiness because we want it to get through that greasy hydrocarbon-rich tail within the cell membrane that I mentioned earlier, but also it needs to have a certain amount of water solubility, hydrophilic character we call this. So lipophilic means fat-loving, hydrophilic means water-loving. Affinity for water versus affinity for grease. And essentially every marketed drug that we take orally needs to have a bit of one of these two properties. If it's too water soluble, it won't get absorbed well. If it's too grease loving, too lipophilic, it also won't get absorbed well. Now, the way we assess this historically, one of the classic methods we've used is a so-called octanol water partitioning assay, and this gives rise to a number we call the log P. Don't worry too much about that, but essentially it's done in a laboratory We now do all this online on a computer, but traditionally, and to do it best, we do it in a real lab, in a real experimental system, where we take a flask, it might be a glass container, and we put 2 layers of liquid in it, water and octanol. And octanol is just used as a sort of a model organic solvent, and it's trying to reproduce that greasy hydrocarbon-rich tail of the fatty acid within the cell membrane. Okay, so these 2 liquids don't dissolve in each other, so the octanol floats on top of the water. And essentially, if we add a greasy, fat-loving molecule and we close the flask, give it a good shake, make sure the drug is well dissolved, and then set it on the bench and let those 2 layers separate out and come to equilibrium, then we measure the concentration of the drug in both layers. And if it's a grease-loving lipophilic drug, then it's mainly going to be up there in the octanol layer. But if it's a water-loving hydrophilic molecule, it's mainly going to associate with the water layer. Now, it turns out that to get a well-absorbed drug with good oral availability, it needs to have both of these properties. Not too much greasiness, but some greasiness. Not too much water-loving character, but some water-loving character. If your drug is at either of these 2 extremes, it's just not going to get across that cell membrane too quickly. Okay, so medicinal chemists are very good now at tweaking the chemical structure of drugs so that they can become a bit more greasy or a bit more water-loving, depending on the needs of the molecule. So this is a real art as much as it's a science, and it's very important in getting both these properties properly balanced in the drug. Another reason that again relates to the fundamental chemical properties of the drug molecule depends on whether it carries an electrical charge, and we don't need to worry about this too much, but certain atoms on molecules in drugs can carry a positive charge, such as a nitrogen. An amine group can pick up a proton and become a protonated charge-carrying species, or if it's a negatively charged oxygen, it can also be charged in duodenum contents. Okay, but the problem with that is that if a drug carries a positive or negative charge, it's not going to get across that membrane bilayer very well. And instead, we want neutral molecules. They're the ones with the best chance of being absorbed across the cellular membrane. So generally speaking, we try to balance out the charges on molecules so that we can achieve a neutral state as much as possible. And it's when drugs are in this state that they have the best chance of being absorbed orally. Another problem that's particularly important for many marketed pharmaceuticals, so now we're moving from drugs that are sort of being optimized by chemists before we ever start using them in humans. For those drugs that we do use in humans, there's often factors that account for low oral availability, and one of them is the fact that those cells that line the interior of the intestines, those enterocytes, make a lot of enzymes that convert parent drugs into what we call metabolites. And I think I mentioned this earlier. These enzymes chew up the parent drug by sticking additional atoms on the molecule that make it more polar and more water soluble and easier for the kidneys to remove. Okay, so this is bad news because you see that it might mean that most of the molecule that enters the body is actually the metabolite rather than the parent drug. Okay, so this is a classic case of why drugs might have a low oral availability if they get extensively metabolized in the gut wall. We're going to devote a whole episode of this series to metabolism because it's really fascinating, but it's also important in using drugs clinically in patients. A lot of problems arise at that level of metabolism. Now, just to finish then, the last factor that we now know is important in determining whether drugs have good oral availability is whether they're substrates for what we call drug transporters. And I like to use the analogy of a molecular turnstile to describe what's going on with these. Whenever you go to a major train station nowadays, you often face this battery of turnstiles, and you have to swipe the turnstile with your train ticket, and hopefully that will recognize you as a legitimate passenger who's all paid up and allow you into the concourse or into the train station. In the same way, cells have all sorts of pumps sitting in the cell membrane, and they have a lot to do with whether or not drugs are allowed to permanently enter into a tissue or get into a tissue. Okay, and these are particularly important. So these are tiny protein structures that are very important at controlling where drugs go within the body. And I'm just focusing here on their role in the gut. But we now know that for many drugs, the reasons they have low oral availability is that these little pump guys recognize the drug after it diffuses passively into the cell, into the enterocyte, and these pumps have sort of a reaction. Well, they recognize the presence of the drug and they think, you're not supposed to be here, mate. Go back where you came from. And so it basically effluxes the drug molecule back into the intestines. Okay, so we now know, and that means that there is less of the drug making it into the bloodstream and more going back into the intestines. Okay, so this is now known to be a critical factor in why some drugs don't have very good oral availability. We now know a lot about these various pumps, where they're present in the gastrointestinal tract, and the sort of drugs that they don't like to allow to enter the body. Okay, very important area of modern pharmacology, and we'll come back to these transporters in some of my future videos as well, because they have a lot to do and to control the fate of where drugs go around the body and whether they get into your brain, all sorts of things. These transporters are there to protect us. Okay, so how do we go about problems with low oral availability when we're developing a new drug if we find that it's got low oral availability? Well, there's a number of things we can do. Most basically, perhaps if we really wedded to that molecule, the chemists, formulation chemists and pharmaceutical chemists can go away and try to see if they can pack the drug with different types of excipients or packing materials to see if we can overcome the deficiencies of the drug to achieve better oral availability. If those things don't work, then it may be necessary to go the tougher route and do a redesign, send it back to the medicinal chemists who redesign the structure of the molecule to improve its solubility, make it more greasy or less greasy, more water soluble, less water soluble, whatever the needs are, or to reduce its vulnerability to metabolism. So this is a more complicated, time and resource consuming approach. So we always have to determine which is the best approach and which is necessary to advance a particular drug. Okay, I've covered a lot of ground in this episode, but hopefully I've given you a bit of an appreciation for how important oral absorption of drugs is. And obviously we want to use the oral route because it's the most convenient, but also the safest way of getting drugs into the patient. We've seen that when assessing the effectiveness with which drugs get absorbed, we often like to use this concept of oral availability, represented by F oral, which is simply the fraction of the dose ingested that enters the bloodstream from the gastrointestinal tract. And then we've seen that if we are encountering problems of oral availability being too low, then there are things we can do to perhaps improve it. Perhaps the molecule might be too big, so the chemist might be able to trim it down a bit, take off some of those bulky groups that make the molecule too heavy. They might maybe it's not greasy enough, so they can tweak the solubility, whether it's soluble in water or grease, to see if they can improve that fraction of orally absorbed dose. Or they might find it's too electronically charged, so they might need to alter some of the parts of the molecule that carry a charge within different pH environments. But most importantly, for most drugs that are marketed as pharmaceuticals, it's often metabolism or transport by these tiny molecular transporters, these molecular turnstiles that have a big role to play in limiting the amount of drug that gets across the gut. So these are really big issues in the pharmaceutical industry. These protective mechanisms of the liver and the gut are very important from the point of view of protecting humans against ingested molecules, but they're sort of the bane of the pharmaceutical industry because they represent functional barriers that make it tougher to get drugs into the body, and so they represent challenges from a scientific point of view if we're wanting to get particular drugs past those barriers. Okay, well, thank you very much for watching. In my next episode, we're going to look at the all-important process of distribution. Distribution is that second process within that four-letter acronym that controls the pharmacokinetic fate of drugs, where they go within the body. Distribution is all important because it's now that determines where the drug goes, whether it actually reaches the brain or the heart or the spinal cord to produce whatever effect we want it to. Thanks very much for your watching. I hope I see you in a future episode. Take care. Bye-bye.